My name is Shana McDevitt. I'm the director of sequencing at UC Berkeley. I've been at UC Berkeley since 2015. I left briefly during the pandemic in 2020, and now I am back because I just missed working with all of the students and missed playing with all of the beautiful new toys and technology, and who wouldn't want to play with the new Revio? If I could give one piece of advice to a new core director or a core director who was looking to get into long-read sequencing with PacBio, and I could only give you one piece of advice, my piece of advice to you would be to focus on QC. And QC, QC, QC. The most important aspect of all next-generation sequencing, but most importantly PacBio, is quality control. Invest heavily in whatever piece of machinery that you need to make sure the sizing on your high molecular weight DNA is correct, the quality is intact, and that you have accurate quantification. And that goes for your RNA as well, because there is no point in doing full-length RNA sequencing if you are not actually generating full-length RNA because it's degraded before you started. And that goes for your DNA as well. Just because we are making 18 to 20,000 base pair DNA molecules does not mean that you can start with degraded product that is nicked and contaminated. You will not get good product. What you do not want to do is plow forward, sequence that, get bad results, and then have to have that conversation with the user that you are charging for something that you should have rejected in the first place. So learn how to QC your samples, spend less money up front, say no, and learn how to do some extractions. The biggest pain point in my workflow is QC. Quality control. It is really, really hard to generate high quality, high molecular weight DNA. We work with exotic, strange organisms at UC Berkeley. We are not extracting things from cell cultures. We're not extracting things from blood. We are extracting insect legs. We are extracting whole insects. We are extracting centipedes, fish gills, livers, lizard tails. These are organisms that are really hard to extract without protein contamination. That contamination continues to degrade DNA even in the -20. It's insane. So trying to work with users and teaching them how to do DNA extractions is harder than doing them myself. So I spend a lot of time doing DNA extractions for UC Berkeley users for free because I want people to succeed. And it is a lot of time because I do old school phenylchloroform extractions because I want 100% success, and that is 3 days of effort. So, QC. My reaction to Spark when I heard about it was initially a little bit negative, but I'm going to explain. As a core director, you have to understand I have a little bit of PTSD from the worlds of the NovaSeq S4 lanes when we went from pooling, 25 to 35 libraries in a lane to 384 to 960, and having to deal with, all of the upfront work to get things together. And so adding sequencing depth and a smart cell for PacBio that isn't necessarily always needed for, say, my insects, felt a little bit like we were going in that direction. And so I had some PTSD about that. And then I had an experience with a user from the University of Hawaii who I actually had been sitting on 2 fruit fly samples for a project of his for at least a month and a half, because What I had received from this user were 2 samples that were absolutely beautiful high molecular weight samples. The ratios were perfection, pure, but I only had maximum of 1. The highest of one of the 2 was 300 nanograms. This was pre-sheared. And before SPRQ, even if I was able to prep these super low libraries, there was no way that I was going to be able to sequence them. But once SPRQ came out, there was a hope and a pipe dream that maybe if I was able to prep these libraries, I would at least be able to load them. And that's exactly what happened. And we got the absolute most beautiful SMRT cell with 2 pooled fruit flies. Not inbred, not pools of fruit flies, just one single fruit fly extracted per sample, pooled, sheared, and prepped on one SPRQ cell. And I believe the throughput was like 110 gigabases with our insert size at like 21 kb, because that's generally like what we shear to, because I demand perfection. So now we're opening up, a by the sample PacBio queue for RNA-seq and for smaller genomes that just do not need an entire SMRT cell, because that's just how the world goes, and you adapt. If I were to encounter another core director that was still using the pre-SPRQ chemistry and was hesitant to move to SPRQ, I would ask them what metrics are holding them back, because I do not understand what metric possibly exists that could be holding them back. I would invite them to just have a conversation and talk to me about what their concerns are, because I'm just not seeing it. Some of the more unconventional species that we have sequenced in my time at Berkeley, so going back from the beginning of our days in PacBio, we started PacBio at the sequel level. We did not go into, we didn't start with the RS systems, but my first project on PacBio was actually a sturgeon, which, if anybody's aware, sturgeon is actually quite a large polyploid genome. And I was asked to do high molecular weight extraction for this project and was provided a desiccated fish fin in an envelope that had just been sitting on someone's shelf for I have no idea how long. That did not work. Eventually, they did have to go fishing with a permit and get some biopsies of a sturgeon, as it is not legal to kill a sturgeon. They're protected species. But that was the beginning of PacBio for me. We have since done the penis worm. I've done some coral sperm, by the way, which plume, or I guess ejaculate once a year into the ocean with someone in Australia. That was a very exciting project. A lot of lemurs right now, lots of leaping lemurs. I am a super big fan of the IsoSeq project we're doing right now, because it's a very large domestic cat project, and I am a crazy cat lady. That's not in any way exotic, but to me it is maybe my most exciting project. It's the cats. The projects at UC Berkeley tend to be very animal, plant related. People who are, the big kind of thing right now is T-to-T genomes. So we are helping people construct T-to-T genomes, or doing fiber-seq, which is a methylation-based sequencing strategy. Not too many plants as of late. We used to be very big in the plant market. And then, of course, metagenomics. There's a large metagenomics focus at UC Berkeley as well. And then some population-based genomics as well. Small amounts of RNA sequencing, now that we can actually do some differential gene expression using the Kinnex IsoSeq strategy. We are starting to see an influx of that growing, but also just the genome annotation for the T-to-T genomes, or even just non-T to T genomes. I mean, for insects or just all of the fish in the world that we sequence, and primates and other just types of more exotic animals. Now that PacBio can call 6MA with the additional upgrades in SmartLink with the SPRQ chemistry, I opt to always provide methylation data for any genomic sequencing that we provide. And it's actually pretty exciting. I always go in and I look, and there's always the percent 5MC and the percent 6MA, and it's noticeable to me in all of the data. And I'm actually now incorporating that in project consultations, whereas, just a few months ago, I was telling people you can't call that with PacBio, and now after reviewing projects just over the last couple of weeks where I'm seeing it pop up in my face, I can now go through and tell people, no, you can call this information. We had a huge grasshopper project that I was talking to somebody about, which was really cool actually, where PacBio was not on the table because where we couldn't call 6MA, but now that's a potential for future projects where that is an issue, and I think that does bring a lot back, potentially for plants and other methylation projects. So it's not just short read methylation projects anymore. So I think the best way to enable utility of your PacBio Revio or sequels is not necessarily to bring in high throughput automation to make all of the libraries that you can. If you have that kind of capacity, then great. But the majority of PacBio cores don't. The majority of us, I believe, likely would benefit from educating and enabling our user base and helping our user base to create their own libraries, their own HiFi libraries, not just Amplicon libraries, and working with companies out there that might have creative tools to help our user base to create libraries that they can then send to Revio owning academic cores. Just like we do for Illumina libraries. If I had 24 hours of open access to the machine to do whatever I wanted, I would like to attempt to open up the Connects protocol and see if I could load other things into the arrays rather than just the 3 packaged kits that are currently available, because I think that the application, it just scratches the surface of what can be done with those arrays.